I've asked everybody to do is to try to, stay around 5 minutes in their presentation. Since we were virtual this year, we opted to do some PowerPoints, so we'll have some visual things to show you. I believe last year we started at the end of the alphabet, so this year, if that's all right with you, Annie, we'll start with you. So I'm going to go ahead and share my screen. And I'll pull up Annie's presentation. And then we'll have a few minutes, for discussion or questions as people would like. So let me pull this up here.
Well, hi, everybody. Thank you for your time and, well, especially your interest in this nitty-gritty stuff. But I also want to thank Hillary Sandler from the bottom of my heart, because without her unfailing support, allowing us to get through the pandemic rules, we would never have gotten any of this done. So as always, Marty and I had a great time, and she will also be speaking, putting this together. And what we realized is this interesting thing that there's always something new. It's like the insect assemblage and the pest issues are always changing. They're completely different from when we started, or even from 5 years ago. And I think the reason for this is that if you think of insect species having this array of factors that regulate them, they're the abiotic factors and natural enemies, we always work on strategies that will be selective and that will study this web of interactions on a species. But what we always are reporting is kind of this whack-a-mole business, where there's a sudden outbreak of an invasive species that probably has escaped its natural enemies. And we also have insecticide-resistant ones like cranberry weevil, which we just don't have alternatives for. And so what happens is this leads to broad-spectrum insecticides being applied, and that may disrupt the web for other insects. So in the next slide, we see one of the worst culprits.
Let me see if I can do that. Hold on for me 1 second.
Scale.
There we go.
But Hillary, I'm down to 20 seconds. One of the worst culprits is Putnam scales. You can see that they're coating the vine. They just reach huge numbers and cause dead spots. And we did a spray trial using a dormant oil, which we see as a very targeted, environmentally sound option. And the data show that when we use the high rate, which is this center area here, this center, if we look at this, the percent dead females, at the high rate, we had the majority of females that were dead. What's interesting in this, though, is that we also had high parasite. We saw that there was a reduction in a tiny black wasp that is an important natural enemy, and probably because they're specialists, they're reducing in numbers as well. But realistically, it may be difficult to get this pesticide or this option, this dormant oil, registered. And also, it may be difficult to get it adopted by growers because you'll be sloshing this oil into the chemigation system. Another outbreak we have is in the next slide, which is a fascinating, tiny, tiny, tiny beetle that's just been ramping up in numbers and causing dead spots. You can see that in the slide there. And what's interesting about it is the mother encases the eggs in feces. And then when the babies hatch out, the immatures hatch out, they encase themselves in feces also. And so it's called a case bearer. Pretty euphemistic because the case is made out of feces. And it's just exploded in numbers. There's hundreds in the sweep net. We sent it to the USDA in Washington, and we got an identification. It is found along the eastern US in small numbers, though. It's not a pest elsewhere. And because there weren't that many of them, ours are now in the Smithsonian collection as well. Bottom line, though, we're using a broad spectrum, harsh insecticide for management. So again, you can see that we're going to have the breakup of the web again because we've gone that way. I think that the last slide that I'm going to do is about my continuing health of bumblebees in the cranberry bog. I put out sentinel hives, and we look at the pollen loads and send them to chemists for analysis, and the chemists are finding high levels of pesticides, even ones that were sprayed weeks before the vines went into bloom. And so they're just, the plants are taking them up, and only 2 of my sites did well this year. So this is going to be an ongoing program for us to figure out how we can get bumblebees to thrive on the cranberry bogs as we have all these disrupted webs. Now Marty's going to talk about cranberry weevil, which is next, where we have a dreadful broad spectrum that we're trying out. Marty, are you there?
It's true. We went out and found our usual 20 or 40 weevil out on state bog, or if you look at the numbers in my thing, it's only, 5 or 10. But I did get a field trial out. It almost killed me. This is my son Corvus, after I made him do the sprays because we were at high gallonage and I could physically not carry the spray tank, and he was pretty exasperated too. Thank you, Hillary, for getting him allowance to be there because we were being COVID committed. The spray trial were danitol and fanfare, 2 pyrethroids, which we've been trying to avoid, like the pox, because they're not good in water, but they have low mammalian tox. Interestingly, the one that is labeled danitol, although not allowed by the handlers, didn't do a thing. It was about equivalent to the control. And fanfare, which has been coming down the pike through the IR4 system for 4 years now, we've already done the residue trials, worked pretty damn good. So we're going to push that through. We've been working with the company. We're trying to force a label out, and then we'll see if we can get MRLs in place. So what a problem. Next slide, Hillary. So our complicated, constantly changing issues continue. We finally got a hold of winter moth with the parasites and with time, and there's just not a lot of winter moth. But this year we thought we had winter moth, but it turned out to be green spanworm. These green worms in the middle, you cannot tell the difference. So I had been rearing them out at my house in my lab kitchen, and these pupa turned into green spanworm instead. And the other thing is, Ann and I published 20 years ago saying that they fly in July. Now they're flying in June. So I got a dozen calls from growers. What are these moths all over our bog? Even state bog, we had lots. The other issue is, as we transfer from Avant to Octara for weevil, Octara doesn't hit any of the leps. So these guys get through, and all of a sudden you have a different problem that you hadn't planned on. We're over 1 minute. Your turn, Hillary.
You are.
But not for
Do you have
lack of
another slide?
trying.
Nope, that's it.
Okay, that's great. So any questions, I guess, for Annie and Marty? I'll do a stop share.
I was only going to say it gives Marty's presentation gives new meaning to taking your work home.
I had those leaf beetles all over my kitchen.
No doubt.
And they're this big.
Thank you. I mean, these are really exciting presentations for those of us who are office creatures.
Yes.
To get to see why we support something like the station, so that excited scientists get to do their work, and we know, for the sake of the growers out there, they followed much more than the rest of us about some of the discussion we just heard. So I just want to thank you both for that work.
Just for the record, some of us are waiting for the good news.
Oh, the bumblebees did well on.
Thank you.
Question, if I may.
I'd like to give my hats off to all the folks down the Cranberry Station too. Insects they seem to come out of nowhere, or like overnight, and it's not something that that the station has a lot of time to really ponder. It just we need decisions quickly, because insects like frost can devastate a crop really, really soon. So it's great to have a relationship at the Cranberry Station and having boots on the ground fairly quickly to ID this stuff, because it means a lot to us, especially with the pressures that we're under. So thanks a lot, guys and gals.
A question.
I think the commissioner had a question.
Thank you. for the entomology team. So we have a new exotic invasive moving up the East Coast, the spotted lanternfly. You know, the states where it's in, they don't grow cranberries. So we've heard it getting into some ag crop. Any thoughts on if this is going to present a problem to cranberry?
Marty and I are both thinking about this. We'll be talking about it for a half an hour at our January meeting to all growers. It's been found in New Jersey, cranberry. So we're getting ready for it. We've studied it, and it is in Massachusetts, up north of Boston, and we're going to be monitoring for it. So it's right on our radar screen. Another outbreak insect. It's in just vast numbers down in Pennsylvania. So it's an outbreak insect introduced from elsewhere.
Well, it's troubling to hear that the New Jersey cranberry crop is seeing it. The only other thing I wanted to quickly mention, the pesticide board subcommittee is going to hold a public hearing on the findings of a scientific literature review that was done relative to neonic insecticides. That's on December 10th. I'm going to put the meeting notice in the chat. Folks might be interested in reviewing those materials or providing written or oral comments.
Okay.
Thank you, Commissioner. Other questions for Andy or Marty? All right, I'll bring up Peter because he's Jeranyama. He'll be next. Look at that. Peter only has 4 slides. What a good man.
Well, you originally said 3, so.
I know. We said 3. Marty beat me up and said 3 to 6. So,
Hey,
but it's all right.
Thank you, Hillary.
Sure. Come on in. This is Dr. Peter Jeranyama.
Thank you. Mr. Peter Baton, he really set me up very well. He said, if a frost can cause you to lose an entire crop. So one of the major projects that I do is on frost monitoring, and I work with the frost committee of the CCCGA. And at issue there is when do we start monitoring for frost? And we've been using 3 models to tell us when to start to monitor, and it's based on growing degree days. So growing degree days is just a matter of taking your maximum and your minimum, you divide by 2, and then you subtract the best temperature for cranberry. The best temperature can either be 44 if you're using the D model developed by Mr. Demoranville, who used to be the station director before he had daughter. And then we also use 2 models from Wisconsin that uses a growing degree day of 41. So the idea is if you're using the D model, once you've accumulated up to 100 growing degree days, you then start monitoring your buds for any possible frost damage. In the picture that you're seeing there, these are sliced buds that we slice under a microscope to see if there's any kind of damage that occurs. So I've got a scale of 1 to 5, 1 being a perfect bud, and 2 where we have 2 floral initials that are damaged, that browning areas, and 3 where you have most of your vegetative initials are damaged, and 4 where you have both floral and vegetative damaged, and 5 is where the bud is tossed, is completely dead. Next slide, please. So this year, our challenge really was that the D model and the simple Wisconsin model, they were not at par with the phenology of the crop. The fact we did get our 100 growing degree days at a time where the buds were not, they'd already advanced so much to the extent that we couldn't use those models, but we ended up using the complex model that was more in tune with the crop development. And the slide here shows the level of damage that we observe under a microscope of various cranberry cultivars, Crimson Queen all the way to Stevens. Anything green means there's no damage. So for the most part, the growers have been doing a good job of protecting their crops against frost damage. Anything red, it means the buds are completely dead. and blue and gold, it's 1 or 2 floral initials that I did. But as we know that cranberry has about 5 to 6 floral initials. So if you damage 2, it means the other 4 are going to initiate flowering for the production of the berry. Next slide, please. So even before I talk about this, I've been also working with CCCGA on developing new frost formulas for the early part of the spring, when to predict when frost is going to occur. That is from April 1st to April 14, and also on the latter end of the season in the fall, November 1 to November 15. We've now come up with the formulas, but now we're at a stage where we have to verify using real data and taking the temperatures on the bog versus the variables that went into developing those formulas. And so far, it's looking reasonable. The other project that I'm involved in is plant nutrition. So I'm looking at nitrogen-rich fertilizers versus the second generation hybrids, because most of our fertilizer recommendations they were based on either the natives or the first generation cultivar. The first generation hybrid is Stevens. So what I'm showing here is the net benefits that you accrue by moving from the recommendation, which is around 40 pounds of nitrogen per acre. So if you go beyond that, do you get any added benefits? Because it's one thing to get a higher yield, but at the same time, your returns you must be able to justify your total variable costs. Are they in tune with what you're going to get ultimately as a profit? So those are different cultivars, and I've been doing this for the past 2 years. And hopefully by next year, we should have good recommendations on that. Next slide, please. I just also wanted to comment on the rainfall patterns for this year. What I'm showing there, the red bars, rainfall, monthly rainfall received in 2018, and the green are for 2019, and the blue are for 2020. As you can notice that the blue, say if you look at June, July, August and September, the blues have been lagging behind the other years. So this year has been quite a difficult year in terms of precipitation, to the extent that some growers, they couldn't even water peak because they didn't have enough water reserves. And some were even failing to protect against frost in the fall because they didn't have enough water reserves, just because we didn't have enough precip to help us out. I think with that, I will take any questions.
All right, so I'll stop my share. If anybody has any questions for Peter about frost or nutrition.
I think I do, Hillary.
Come on in, Steve.
Steve Ward. I had probably my worst crop in probably 10 years this year. I'm trying to kind of zero in on it, whether it was frost damage or not. I'm wondering if the winter being so mild left our vines somewhat more stressed than normal, having not been underwater, or did they ever go dormant enough? And could that have been the problem with my terrible crop?
Yeah, I think what is at issue here is the physiology of the plant, because when you have those mild winters, you sort of like have a stop and go in terms of dormancy. Some of the buds will sort of like break out of dormancy because the temperatures are high enough, and then it's followed by a cold spell. So you tend to have more bud damage in that instance. Whereas if it stays cold, the buds will not break any dormancy at all. So I think, and we did call quite a bit, a record number of frost nights in the spring. Yeah. It was quite something. And fortunate enough, although we couldn't meet in person, we're doing it on Zoom. I'll be on State Bog, and Anne and others will be at Rossbrook, and we'll compare pictures on Zoom, and then we'll come up to a position where we say, oh, it's a 27 tolerance, and so on and so forth. But it was quite interesting to be doing it that way.
Thank you.
No, I think Peter brings up a good point. You know, some of our extension activities, being with COVID, it kind of pushed us into new realms, which maybe we went a little bit kicking and screaming, but in some cases, it really ended up being, you know, quite a nice venue for, you know, multiple people to be able to see what the bud status was in the spring. And I'm sure that a lot of these techniques will carry past COVID. So that was really kind of a nice outcome from that. All right. So the next we have is Giverson Mupambi. So hold on for me a second here, Giverson. So Dr. Mupambi is coming up in January. Will actually be his second year with us here. So one of our newer faculty members. So go ahead, Giverson, jump on in.
All right, thank you, Hilary. Hello, everybody. I must say this was a really interesting second year for me. I was hoping to start ramping up on my research program. Next slide is Hilary, but obviously with the challenges we had, I couldn't do some of the research that I intended to do. Next slide.
The next one
So
after
in terms
this?
of, no, this one is fine.
Okay.
So in terms of my research program, I had 5 major research programs. First thing, I was still continuing my research on the use of plant growth regulators, and I was diversifying that research into looking at different aspects. So I was looking at improving fruit quality, improving fruit set, controlling the vigor, and then also improving canopy establishment after bulb renovation. Then I was also continuing a project that we had on canopy management, where we're looking at mowing and pruning to improve fruit quality. So last year we had really encouraging results, but this year we're trying to look at the recovery of the canopy. And then I also had work where we're looking at biostimulants to improve fruit quality. This is part of our sponsored research that we had. We also had a project last year on solar research that we hoped to continue this year, but the construction of the projects was postponed because of COVID. But hopefully next year we'll be able to do that. And then we have a new project that I'm really excited about, where we're looking at the evaluation of new cultivars. So this is part of our new H project. And we also have funding from Northeast to look on that. So we are looking at especially new varieties that might have improved disease resistance, fruit quality, and yield. As you are aware, we are losing some of our pesticides that we have. So if any of these new varieties could be helpful to us. So we have varieties from Valley Corporation, Rutgers, and University of Wisconsin. And then some of these, especially the ones from Rutgers, have been bred for fruit rot resistance. So hopefully we'll be able to, in the future, to have some new varieties that offer improved fruit resistance to our growers. Next slide. So in terms of results, I've got a couple of slides that I'm going to show you. So my work is mostly fruit quality, and we're currently really busy in the lab now, so we're still synthesizing the results. But in terms of the fruit color, this was the second year that I was doing this work. So last year I was looking at plant growth regulators, but I only had a protein, which is abscisic acid. And this year I was able to expand my program. So I had Ethefone, 2SO, and Motivate. Those are 2 ethylene releasing compounds. And then we had Blush, which is a jasmonic acid compound, and then Sandler, which is an oxylipine, and Proton. We also repeated with Proton last year, but at a higher concentration. So what we did this year was different because we did it with the recommended adjuvants, but we also looked into different adjuvants, especially adjuvants that might increase penetration. Because last year we didn't get really good results. So we included Hebimax, which is an oil surfactant, and then MSO concentrate, which is a methylated seed oil, and Veda, which is a proprietary adjuvant. But all the benefits of these adjuvants that they have increased penetration into the canopy. So if you look at this graph that you had, this is the data that we look at, where we look at the combination of these plant growth regulators with Veda. As you can see, some of our plant growth regulators perform very well. So when you look at Motivate, which is your Ethefone based, your ethylene releasing compounds, we're able to increase So if you look, we are looking, this bars shows class 1 to class 5, whereby class 1 is green fruit and class 5 is like your red fruit. So we're able to have over 97% fruit above class 3 in one of the ethylene releasing compounds compared to the other 4. So I think this pushes the idea that you might need to have different adjuvants, especially with plant growth regulators. Especially last year, we had disappointing results. So I'm really excited about this. I'm to have other adjuvants that I need to look into, but this is really exciting for us. So in order having us a cultural practices for improving fruit color. Next slide. Next slide.
It's there.
Okay.
This is not the one you want?
Yes. I think my internet is slow. Yeah, that's
Okay.
the one, yes. So one of the things that we also looked at was a fruit set. I think this project idea came from Jeff Capel. So Jeff, this is a project for you. Last year during the meeting that we had, the summit meeting, as if we could look into fruit set. So I went and thought of how we can improve fruit set in Cranberry. So I looked at 2 compounds. We looked at Splendor, which is a cytokinin, and also looked at ProGib, which is gibberellic acid. So the good news is we were able to increase fruit set. So if you looked at the graph to your left, when you look at the number of berries per square foot, they were significantly higher, especially with ProGib. So the gibberellic acid treatments, they were able to increase the fruit set. But the bad news, however, is that the vines were not able to support the additional fruit set. So if you look at the fruit size, which is a graph to your right, you're having smaller fruit. So overall, even if you have more fruit set, you still have smaller size fruit, which means that your yield is not changing. I think the pictures, if you look at the pictures below, it shows some of the vines that were treated with gibberellic acid. I mean, you can see there is a lot of small size fruit, and this is almost close to harvest, a week or 2 before harvest. So I think next year when I do this research, I think the idea is to look into what can you combine with fruit set. So if you want to increase fruit set, maybe improve your nutrition so that you are able to support these additional berries that you have. But this is really exciting for us because if all this additional fruit that you're able to set, if they become full-size berries, then we might find a way of improving yield. Hilary, thank you. I think I'll end here, Hilary.
That sounds great. So I'll go ahead and stop share. So I think one of our exciting outcomes that we had for this year was the use of Zeus, which is a pre-emergence herbicide. A lot of the work that Katie has been doing over the past few years has developed some crop safety and some efficacy data. We were able to get a special local needs to apply this compound. It has to be applied before cabbage head. Otherwise, you get a lot of cranberry damage. But moss has been an increasing problem on cranberry bogs, primarily this hair cap moss, which almost kind of looks like little pine trees when you see it in the field. But here we did apply it on state bog because unfortunately, we have a huge moss infestation on several of our sections. So we did put it out there, and it is a little bit slow acting, but you can see the great efficacy that we had, even though some of the tips of the moss are starting to green up. The big thing that we notice is that the mosses produce a lot of sporulating features, and those were very much reduced as well with the use of Zeus. So we hadn't done a lot of work on sphagnum moss, but some of the growers did try it. And so here you can see an untreated area where the sphagnum moss is very happy and green. And here's an area where we did treat it. And again, the moss was looking very, very unhappy, which as weed scientists, we're always very excited to see unhappy weeds. And here again, another issue, of course, with putting out any pre-emergence during the spring season is that it always overlaps with our frost season. And there's this constant balance between trying to get the pre-emergence herbicides out and not having them then be subjected to 8, 10, 16, 20 hours of frost events after application. But as it would have, as Peter had mentioned earlier, this was an incredible frost season. And so the Zeus was applied on April 16th. We then had 8 hours of water that night, 6 hours the next night, and then rain. But we still were able to show quite a significant impact on the moss. And this was sustained throughout the season. So we were very, very excited to see this product. It has a distinct mode of action compared to the rest of herbicides that we have in our toolbox. So from a resistance management point of view, we're very excited to be able to have this compound. Some of the other work that we've been doing is part of our Hatch project, which is some federal funding that's administrated through Jody's office. This is actually with all the faculty. We each have our own part in this. And Katie and I have been looking at the herbicide tolerance of our new large fruited varieties, which you heard Giverson speak about. So we were looking at Crimson Queen, Demoranville, Mullica Queen, and Stevens. And I don't really have time to go through all the different treatments, but we basically put out all of our herbicides in different rates and fashions. And we did see some yellow vine syndrome showing up, which is this is an unnatural yellowing of the leaves. And it usually has like a little Christmas tree. You can see that the central portion of the leaf remains a little bit green, but everything else is yellow. And this has been associated with casseron use, but work that was done by Bruce Lampinen, which was a few physiologists ago, he was actually able to demonstrate that it really has to do with water imbalances. And then when you apply an herbicide such as casseron, which inhibits root formation, this really exacerbates the stress on the plant and really disrupts the water uptake ability. And then we end up getting this yellow vine syndrome. So with these particular plants here, we had applied 60 pounds of casseron 3 years in a row. And as you can see, 2 out of the 3 years, we did see symptoms. We saw it across all the varieties, again, exacerbated by drought, which could certainly come to be a concern with us. These situations happening more and more with climate change. It does tend to show up late in the season. There's not much you can do about it. But we did follow this up with yields, not only in 2016, 17, 18, we also have 19 and 2020 data, and we're not able to correlate any yield impact. So this looks terrible, but doesn't really end up translating into anything of a yield difference. The other symptomatology that we did see was this Callisto tip flashing. Callisto is an herbicide that interferes with the photosynthetic apparatus in the plant. So typical symptoms are this blanching that you see here, and growers, and we have seen this tip flashing where after you put the Callisto on there, the tips become white, but then they tend to grow out of it. And here you can see our treated plot right here, how you can really pick it up from the road as you drive by that there's some issues going on. So here we did see some yield impacts, but again, when you're using these concentrated applications of Callisto, they're usually very much spot treatments. And so based on the reduction in the yield, we would estimate that a grower would tend not to see a yield depression because the treated areas would tend to be so small. And then finally, I just wanted to talk about some of the work that I'm doing in conjunction with Dr. Annika Saito and Jacob Scott from the biology department. Again, this is work that's been supported by CAFE's Integrated Research and Extension Project. And this is capitalizing on a lot of the expertise of Dr. Casaido. She has spent quite a bit of her time looking at some of the genetics of weedy rice. And I was able to attract her to thinking about looking at weedy dodder. And so we have collected dodder from commercial sites and wild sites And if you're not familiar with what dodder is, it's a parasitic plant. It doesn't produce any of its own chlorophyll. It must attach to a host in order to complete its life cycle. And it penetrates the host with specialized structures that are called haustoria, which then extract all of the food and the water out of the plant. And dodder is just exquisite in the way that it has adapted to cranberry systems and to cranberry, because it just disables the host. It doesn't kill the cranberry, but it does deplete it of enough carbohydrates in order to significantly reduce yield. And you could see if you have this out on your bog, it is a substantial problem. It just looks like somebody went out and threw spaghetti out there. So it covers the canopy and inhibits light penetration, and this decreases fruit size as well as fruit quantity wherever dodder occurs. So we had noticed some phenotypic differences. Sometimes there was this skinny yellow dodder that was out there, and sometimes there was this more robust orange dodder that was out there. And actually, interestingly, the robust orange, you could actually pull it off the host much more easily than you could the skinny yellow. So we were wondering, even though we could see physical, visual differences, did this actually translate into any genetic differences? So we wanted to know actually what species of dodder were out there and how much genetic diversity was present on the farms, and would this be able to inform some of our management recommendations that we would make? And I know that this is kind of a busy slide, but this is the gene map that we were able to construct after sampling about 426 samples from New Jersey, Wisconsin, and Massachusetts. And so I thank my colleagues from those areas. And we also went to some wild sites. So we had always supposed and suspected that we were working with Cuscuta granovii. And in fact, we were able to confirm that most of the dodder on the commercial bogs does seem to be this granovii, and is a very common species that's found in wild sites as well, and is also native to the United States. The interesting thing is that we actually picked up a species that we hadn't heard of before. This was from New Jersey. Corylli was actually in 2 different samples. And so that was very interesting. And then we also picked up quite a bit of Cuscuta campestris, which is a very common parasite on many different crops, including alfalfa and tomato and citrus. But this one seems to be predominantly on our commercial sites and not on our wild sites. So some of the work that we're going to continue doing here is to see if we can figure out if there are any differences between the weedy dodder and the dodder that we find in the wild sites, and that hopefully this information will eventually translate into some management recommendations concerning dodder infestations on Massachusetts bogs. So that was all I had for my research. I did just want to mention, we have had quite a lot of extension efforts that have gone on. We were able to get a Section 18 for the use of curb, which is very important for our growers who do have dodder on their bogs. And we'll also be petitioning to get another Section 18 for the use in 2021. We were able to get that SLN for the use of Zeus for both Massachusetts and Rhode Island, and also in the use of intensity in Rhode Island. So we have a couple growers in the Rhode Island area, and so we've been trying to bring them into our fold whenever we're petitioning for different special local need uses. So I'll just kind of wrap it up with that and be happy to entertain any questions that people might have.
Hillary,
Yes, Steve.
has the dodder pressure reduced over the past few years, or are we just doing a better job managing it?
It has certainly been my perception that it seems like dodder has gone down. It's nowhere near the widespread nature that I first remember, certainly in the 1990s. Now, whether that's, some of the rumor has been that that's related to our reduced use of chlorothalonil, because the thought is that the chlorothalonil as a fungicide has been controlling the natural enemies or the natural pathogens of dodder. And that's why a lot of times we may not see it in natural areas as much, but we haven't been able to prove that scientifically. But it's certainly been my perception that there's not as much around. Oh, he's got a little background noise going on. So can one of my alternate hosts help me with that, please? So, any?
I have a question about moss.
So Linda, I think your connection is terrible, but so if you could just write your question in the chat, I'd be happy to do that.
She did. It's did you get moss on
Okay.
renovated bogs with a clean rooting medium?
I haven't seen that, but I don't know if one of the growers on the call who have renovated bog have made any observations on how quickly moss moves in. I've primarily have seen it on established farms. So I don't know if Peter or
we have observed
John?
it, Hillary, on
Okay.
on renovated bogs.
Absolutely.
You know, and historically, it was generally, if you had moss, it was a wet area. You had to fix it with drainage, but now it's definitely just seems to be growing everywhere along,
Yeah.
you know, dry edges of renovated bog. And I know it's a gaining concern, increasing concern to a lot of growers.
Sure. And I really think that this is, moss must have been a species right on the edge for climatic conditions, because it's not only becoming an increasing problem in agricultural areas, moss is becoming increasingly a problem in residential and across many other types of environments. So that would be one thing that we're going to have to explore is if people could use Zeus on you know, newly planted bogs. We do have some observational evidence that it should be good on new plantings.
We had Peter Beaton, we had a particular bog that we had a traditional Howe farm vine variety. So it had high moss incidence. We put on the Zeus right at the proper timing. But right next door was a very newly renovated piece of bog, Mullicas, and that was in the cabbage head stage. You're correct. You do not want to put on Zeus on cabbage head stage. But what I did notice was that there wasn't a weed on that bog where it overlapped, and where the remaining part of the newly renovated bog took constant attention, particularly from the grasses, and ultimately the renovated vines that were hit with Zeus, they grew like wildfire. So actually it was a good thing, but that was an observation. Yeah. So, and I also noticed that Zeus, it begins with the moss as the thinnest, and then it works into the thickest part of the moss. I just have to have a little bit of patience, but it was probably the eminent material, most important new material that we put on, I believe, this past year, because it did really work, you know. So I'm very, we have a lot of moss, so I'm very happy with that.
We have a lot of moss too, so we were very excited as well. Yeah, it is one of those compounds where you do have to be very patient, like 4 weeks, 6 weeks, waiting to see effects. So, all right. So I think we'd like to move on to Dr. Uppala. And let me share my screen again. And then we'll have a couple things just to mention at the end. I know I'd like to, and I don't know how the chairman would feel about this, but we're kind of hoping to keep it to about 12 o'clock because that is 2 hours for people, but totally up to you,
I think
Chairman.
that would have to be an outside time. I know I have another call scheduled at noon, so
Sure.
not that, but I think that that's a good target as an outside
Okay.
time to conclude.
Okay, so let's go to that because Dr. Uppala is our final presentation, and then we can wrap up as need be. So Dr. Leela Uppala is also our second young faculty member that's here with us, and she just passed her second anniversary with us, having started in late October. So take it away, Leela.
Okay, thank you, Hillary. Good morning, everybody. I'm going to talk about Cranberry Pathology program. There are 4 diseases that have been prevalent and causing significant economic impact in cranberry in Massachusetts cranberry cultivation. Among these 4, fruit rot is the greatest challenge, and it is peculiar in so many ways. For example, upright dieback or Phytophthora root rot or fairy-ring, these 3 diseases warrants attention and management only after symptoms appear in the field and or laboratory diagnosis is confirmed. On the other hand, fruit rot, next slide, please.
Sure, hold on for me 1 second. Here we go.
Fruit rot, a disease complex associated with over a dozen fungi. The fungi attack flowers and remain latent in the fruit, in the flowers, and multiply in the developing fruit, and symptoms manifest as field rot right around harvest time, so which is too late to mitigate at that point, or during storage as storage rot. It has been a persistent challenge for over a century in cranberry cultivation in Massachusetts. And in current industry, where more than 90% cranberries are processed, fruit quality standards are stringent, and if a particular load or a delivery has more than 20% rot, they are outright rejected by the cranberry handlers. And we don't have a predictive framework that allows growers to understand what season the fruit rot might be more and what factors contribute to high fruit rot incidence. So to avoid the loss, the growers are spraying up to 5 fungicides per season, mounting significant amounts to cost of cultivation. Next slide, please. So then, and another problem is increased disease pressure associated with new hybrids or cranberry varieties, which are less tolerant to fruit rot. They're high yielding, but less tolerant, and shrinking toolbox. And growers are used to broad spectrum fungicides like chlorothalonils and mancozebs for the past 4 decades. And in 2019, export markets targeted chlorothalonils, and in 2020, they targeted mancozebs for perceived environmental risks and human and aquatic animals toxicity. So there are more and more stringent regulations on the use of chlorothalonils and mancozebs. So the industry is shifting towards single site mode of action fungicides like group 3 and group 11, which are effective, but they pose higher risk of selection for fungicide resistance. Next one, please. So the immediate short-term research goals are to diversify our toolbox before we see resistance concerns. So I initiated research projects to evaluate novel fungicides with different target site or different modes of action, and also eco-friendly management strategies like canopy management, cultural practices, and biological formulations, with a goal to develop true integrative disease management program. Next one, please. So the preliminary, here are some of the preliminary results. They are promising. We identified some products which are able to reduce fruit rot significantly, and also are under safety standards. And this is not an exclusive list. I have more compounds we are evaluating, and data is being processed. Next one, please. And I also initiated some long-term research efforts with a goal to come up with a better fruit rot prediction model that will allow growers to make informed decisions on fruit rot management strategies. For example, if a particular season or a location or factors associated with a field are putting their field at high risk, then they could spray more, or they can do proactive management strategies. In other years, they could spray less. So I'm hoping to develop a data-based scientific fruit rot prediction model that will allow them to reduce inputs and cost of cultivation and produce high quality fruit. Next one, please. With that, I would like to thank my collaborators and funding agencies for providing funding for this research. I also want to thank faculty and staff at the Cranberry Station for their continued support for the past 2 years, and my technician, Ms. Crystal DiMaranville, and research associate Rayan Jaherling for their sincere efforts towards our essential research. And I also want to thank Massachusetts Cranberry growers, especially John Mason, Rob Rubini, and Matt Beaton for allowing us to do research in their bogs. Thank you so much, and I'll be happy to answer any of your questions.